So today we're going to be talking about protein synthesis. I say that as if I'm going to be making these videos consistently. I am not. Anyways, so protein synthesis is how the cell makes proteins. Protein synthesis is split up into two parts, transcription and translation. Transcription takes place in the nucleus. It starts out pretty similar to DNA replication, where helicase comes in and it unzips the DNA. But instead of DNA polymerase coming in and duplicating the DNA strand and everything, RNA polymerase comes in and it creates a strand of RNA. RNA is short for ribonucleic acid instead of deoxyribonucleic acid for DNA. This specific strand of RNA that it made is called mRNA, or messenger RNA. We'll get to why it's called that in just a sec. So RNA is a lot like DNA, but there's two main differences. First of all, it's a single helix instead of a double helix. So DNA has two strands, and they kind of swirl together like that, but RNA only has one strand. Second of all, RNA uses uracil instead of thymine. So DNA has bases A, T, C, and G, right? That's the whole alphabet with life and everything. Instead of T, RNA has U. Ribonucleic acid just doesn't like thymine, but uracil taking its place works just fine. So when RNA polymerase is making the RNA, whenever it has an A on the DNA strand, instead of putting a T on the RNA, it puts a U. So it does that whole process along the DNA strand, making the RNA. Anytime there's an A, it puts a U on the mRNA. Anytime there's a T, it puts an A. If there's a G, it puts a C. If there's a C, it puts G. So again, basically the same as DNA replication, except a U instead of T. So it's called messenger RNA because it sends the message of the DNA code to the place where it makes the proteins and everything. So once the mRNA is finished, it leaves the nucleus through the little nuclear pores. You know how a nucleus has little holes in the side? It can leave through there. The reason DNA doesn't do this on its own is because, first of all, it could become damaged if it leaves the nucleus. And second, it can't fit anyways. The pores are too small. However, since mRNA is a single helix, it can slip right through. From there, it moves on to the ribosome, also known as rRNA, where it starts translation. So during translation, something called tRNA, or transfer RNA, lands on the mRNA and drops off an amino acid to create a polypeptide chain. So in the language that mRNA and DNA, tRNA, everything uses, there are three-letter words known as codons. These codons are how the tRNA knows which type of amino acid to drop off to make the polypeptide. tRNA has something called an anticodon, which is basically the complement to the mRNA's codon. So on the mRNA, it has special codons called the start codon and the stop codon. The start codon tells it where to start, and the stop codon tells it when to stop. The start codon is always AUG on the mRNA strand. An easy way to remember this is AUG, like August, which is when school starts for a lot of people. So once the mRNA is in the ribosome, it goes through it almost like a conveyor belt in a factory. And it passes three different areas: the A site, the P site, and the E site. So in the A site, a tRNA touches down on the mRNA. The mRNA then moves along by the length of one codon. So now that first tRNA is in the P site. Then another tRNA lands in the A site. The amino acid that the first tRNA was carrying then detaches from that first one and latches on to the one that the second one is carrying. Then the mRNA moves down again. So the first tRNA is now in the E site, the second one is in the P site, and the A site is currently empty. The first tRNA that's now in the E site is going to exit, and it's going to find another amino acid so that when it's its turn again, it can land again and do the same thing all over again. While it's doing that, another tRNA lands in the A site. Then the amino acids that were connected to the one in the P site detach and latch into the one on the A site. Then the mRNA moves over again, and the whole process continues until we have one really long polypeptide chain made of a bunch of little amino acids. Now we're almost done. but it's not quite a protein yet. Once you have the polypeptide chain, it goes to the rough ER, which is on the outside of the nucleus. Inside of the rough ER, it's going to get twisted and bent and curled until that polypeptide becomes a protein. Isn't that super cool? And that protein can be used for all kinds of stuff. For example, if it made keratin, it can be used to build your hair or your skin or your nails. Proteins can also be used to make enzymes, which help speed up chemical reactions, as you've probably learned by now. Protein is also used as energy to help your muscles move. I wouldn't be able to be moving my hands so much as I'm talking if I didn't have protein. I got a little too excited there, I'm sorry. But yeah, so protein synthesis is a really cool process, and it's definitely really necessary. Otherwise, you wouldn't have hair or a skin or nails, and you wouldn't move, and everything happening inside of your body would be really slow because you wouldn't have any proteins. But yeah, that's all I have for you guys today. Thank you so much for watching. Please don't check out my other videos because they are very, very dumb. Other than that, thank you. I just said thank you. Bye!